I'd like to welcome you to today's presentation of the Congressional Biomedical Research Caucus. Before we start, I'd like to thank the co-chairs of our caucus, Representative Steve Stivers from Ohio, Representative Joe Barton from Texas, Representative Jackie Spear from California, and Representative Steve Cohen from Tennessee. The Coalition for Life Sciences has been hosting these monthly caucuses for about 28 years now. But without the commitment and dedication from the chairs of the caucus, we would not be able to offer these briefings and the opportunities for you to hear from some of the nation's top scientists. Today is one of those opportunities that you will have a chance to hear from one of our nation's top scientists, Dr. Matthew State. Dr. State is a pioneering researcher studying the genetics of children with autism. Autism. Now that's a word, the condition that evokes complicated feelings and intertwinedness of lessons and challenges. Parents who have been told, yes, their child has a form of autism, have many questions, but all are rooted in the why and how. There are many theories. I'm sure we've all heard our share of them. But today we are going to hear from Dr. Matthew State, who is a child psychiatrist and human geneticist studying pediatric neuropsychiatry syndromes. And today, maybe we can all learn a little more about the why and the how. Dr. State's lab focuses on gene discovery as a launching point for efforts to illuminate the biology of these conditions and to develop novel and more effective therapies. Dr. State received his undergraduate and medical degrees at Stanford University. He completed his residency in psychiatry and fellowship in child psychiatry at the UCLA Neuropsychiatric Institute and earned a PhD in genetics from Yale University. He was on the faculty at Yale from 2001 to 2013, where he was the Donald J. Cohen Professor of Child Psychiatry, Psychiatry and Genetics, and the co-founder and co-director of the Yale program on neurogenetics. He is currently the Oberndorf Family Distinguished Professor and Chair of Psychiatry at UCSF, and Director of the Langley Porter Psychiatric Institute and Hospital. I know we are all looking forward to hearing you speak today, Dr. State, so without further ado, please join me in welcoming him.
Thanks very much. It was a wonderful introduction. So I'm going to talk today about the genetics of autism, and I'm going to cut to the chase and tell you that by the end of the talk, I hope to convince you that genetics has already transformed the understanding of autism, but that there's an enormous amount of continued work that needs to be done to make sure that the discoveries that we've already made are translated into new and better treatments and affect the lives of the children and families with this syndrome. So this is how I'm going to lay out the talk today. I'm going to first talk to you about what autism is, what autism spectrum disorder diagnosis means. I'm going to give you an idea about why it is that we're looking for genes, and then I'm going to pause momentarily for about 4 minutes to give you a genetics 101, just to make sure that we're on the same page and you understand the lingo if you haven't done human biology recently. So then I can take you through what we've learned so far about the genetics of autism, which genes and sections of the genome have been found that lead to autism, and then ultimately the most important question, which is what have we learned from this, and what can we learn now from the genes that have been identified. All right, so we're going to start with what is autism. And I'm going to show you a couple of videos and watch closely and see whether or not you might be able to make the diagnosis. So this one you don't need, there's really no sound. The child doesn't have useful language. The interviewers talk, but he does not. That's his father and an interviewer. And I just encourage you to take a look both at his movements as well as what you can sense about his behavior and the way that he's interacting with people in the room. He's a 13-year-old boy, and so if you have a picture in your mind of what a 13-year-old boy would typically look like in this kind of setting versus what you're seeing here. Okay, so now I'm going to show you another video, and again, see what you think.
In what ways did you think you were different?
Well, in elementary school, I was always walking around the playground myself, deeply involved in some imaginary game. I would sometimes play with friends, but they would play by themselves. So I would walk around by myself. Back then, I never even really seemed to think what other people were thinking. I never it never dawned on me to think about that. I mean, it never occurred to me that other people were probably thinking something different, and I still have that problem. I still don't really think exactly what other people are going to. I still just find it difficult to think, well, what are they thinking about?
Okay, so if this were medical school, I would quiz you about what you saw in both of those videos, but I'm not going to do that today. I'm just going to tell you that both of these individuals are diagnosed with autism spectrum disorders. And the reason is that despite many, many differences in the way that they appear, they share fundamental impairments in reciprocal social communication, their ability to interpret and convey social meaning in their interactions, and that's accompanied with restricted interests in repetitive behaviors and/or repetitive behaviors. Now, in the first child, if you watch closely, the minute that we turned on the video, he was doing some hand flapping, which is a classic repetitive behavior. We call it stereotypy in autism. The second young man would be harder to tell, but if I ran the interview longer, he would start talking about a very highly restricted interest in trains. So both of those things, again, so you can get a sense that while there is sort of a core set of defining features, in fact, that's how we diagnose autism, that this is really a collection of disorders that describes a broad spectrum of difficulties. And consequently, we've moved from calling the syndrome autism to calling it autism spectrum disorders. So that change reflects sort of a broader transition in thinking about the diagnosis of autism. 30, 40 years ago, only the first child would have gotten the diagnosis of autism. Now both of these individuals would, again, because of the sense that despite the differences, the overt differences in how it manifests, that they're sharing these core sets of symptoms. And this change in the diagnosis has absolutely contributed to some degree to the increasing prevalence. And I'm going to talk a little bit about that. Currently, the best estimates are their autism affects about 1% to 3% of the population. The CDC recently came out with their latest revised figure of 2.24, which is overly precise, but it's about 1 in 50 children would get the diagnosis again in this broad spectrum. So when people talk about the increase in the rates of autism, we know for sure that one of the reasons is that what we're talking about relative to the numbers that we would have cited 40 years ago is a much broader set of conditions. And then the second is that when you take a look at these figures, what you find is the diagnosis of autism has increased. The diagnosis of what we would have called mental retardation, now called intellectual disability, has decreased, and we call that diagnostic substitution. So a significant contributor to the perceived increase in the prevalence of autism are both of those things. Increased ascertainment, or three things. Increased ascertainment, we're better at identifying it, a broader definition of the disorder, and diagnostic substitution. Now, even when you take all of that away, there is some evidence that actually the true underlying rate of autism as we would have defined it 40 years ago has increased over that period of time. And I'm going to get through the genetics and then come back to explain to you how that observation is still consistent with now what we understand about the causes of the syndrome. The total cost of autism to society, these are very broad estimates. The Autism Speaks, which is a major advocacy group, supported a study that was recently published, 2015, which estimates by 2025 somewhere between $500 billion and $1 trillion, depending upon whether you model the current prevalence of autism and carry that forward, or whether you look at the trajectory of prevalence and imagine that that's going to continue to increase. Regardless, it's obviously a significant burden. So that's the backdrop, and now I want to address why, in that context, we would look for genes. So the first reason is that there are literally decades of evidence that genes play the largest role with regard to autism risk. So of all the disorders that we study in psychiatry, the estimates looking at families and how autism clusters, not only within families but among twins who share different amounts of DNA, autism is the most genetic. So more than schizophrenia, more than bipolar disorder, more than depression, autism is the most genetic. Now that doesn't mean that it's entirely genetic, and there are estimates that about 20 to 30% of the liability in the population is carried in the environment. We don't know what in the environment. What we do have good data about is that it's not vaccines. There have been more studies of more children vaccines than of any other environmental factor that might be contributing to autism risk, and there is literally no evidence. In fact, there's evidence that there is not a significant risk to the population with regard to childhood vaccines and the development of autism. Now, a very important reason to study the genetics of autism is that currently what we can do about autism, which is most often, a lifelong condition that leads to significant impairment, is that our treatments are very limited in efficacy. The mainstay of treatment are behavioral interventions. We use educational kind of approaches, behavioral kind of approaches to help kids. There is nothing that targets the core symptom of social impairment. And we use medications, but those medications are generally for the associated symptoms, including behavioral difficulties, et cetera. And again, don't get at the thing that really defines autism for the disorder that it is, this fundamental impairment in reciprocal social interaction. And it's clear that compared to other areas of medicine, that over the last 30 years or so, and longer than that in fact, that it's been a nearly total lack of understanding about why, at the level of the molecule and the cell, and ultimately the brain, that we don't understand, have not understood why autism arises or what the underlying pathophysiology is, what drives it, and as a consequence, our ability to think about how to do a better job with regard to treatments has been severely limited. So the strategy here is that if you can get to the level of the gene, which is you can get to the level of the molecule, that could be one important strategy. We know that they contribute. It would get you to the molecular level, and then potentially you could use that in order to try to begin to pull on the thread to understand what is the underlying biology, why does it happen, and that in fact for 20 years has been the kind of driving motivation in my lab. I'm going to show you just a little diagram, but to drive home a point about sort of why this particular strategy. Now, I've told you that genes are not everything. We are not doing this because we believe that there are no other factors that contribute to autism. We're doing it because it's a particular way to try to get traction on the question about what's going wrong in the brain. So again, this is a child with classic autism, and what we're trying to do is to get to the level of the molecule, to go directly from a complex human behavior to use the most powerful tools that we can to identify where in the genome that there are changes that contribute to the risk of autism. And I have to tell you, when I told folks that I wanted to do this after completing my PhD, that there was a lot of skepticism about the ability to do this. There are 3 billion bits of information in the human genome. We already knew that this wasn't going to be simple, that the disorder, as you've already seen, is very broad. It's quite diverse in terms of the genetics. We knew that it wouldn't be searching for one gene, but potentially, at the time, dozens, maybe more genes. And so the notion was that this was an extremely complex task to sift through billions of bits of information to find the genetic roots of autism. But in fact, one of the things that led me to this is that my estimation as a child psychiatrist and as a human geneticist was that this was the simplest part of the problem, the simplest inroad. And the reason is when you look at the level of complexity of the human genome and you begin to compare it to the level of complexity of the human brain, it is, in fact, the easiest and most tractable data set. So as compared to 3 billion bits of information in the genome, there are more than 150 billion different cells in the central nervous system, and those contribute to more than 100 trillion connections that make up circuits that drive behavior. So if you're thinking about a way into a problem as complex as this, we decided to take a tough but really the simplest approach, and again, to try to move from the understanding, if we could get there, of genes that had a significant effect, and to pull on this thread to get us to the cells, to get us ultimately to the circuit level understanding of why this is happening. And all of this is a prelude then to beginning to understand how we might be able to develop novel treatments that would do better than we do right now. All right, so that's the prelude, and now I'm going to take a step back. For those of you who've had human biology recently, you can take a 4-minute nap. And for those of you who haven't, I'm going to give you a quick 30,000-foot view of Genetics 101. So the first thing I'm going to do is talk to you about DNA, just to review for you. So DNA is the blueprint for biology. It is comprised of 4 major component parts that we abbreviate A, C, G, and T. So the DNA letter code is only 4 constituent parts, again, contributing to this being sort of the easiest part of a very complex problem. Now, among the 3 billion letters in the genetic code in the human genome, which is present in every cell that has a nucleus in your and everyone else's body, there are about 20,000 genes. So it's only about 1% of the genome, and I'm going to come back in the next slide to tell you more about genes, but the things that we're looking for already constitute only about 1% of the 3 billion bits of information are carried in your genome, and the rest is really poorly understood. So what goes on between the genes, and the genes actually help drive forward biological processes. It turns out that everything in between is either not understood, or what we do understand is that to some degree it helps regulate and control the activity of that 1% of the genome. So DNA, as opposed to being a very static molecule, actually is highly dynamic and controls its own kind of biology. And that's for another long talk, and I'm not going to take you any further down that rabbit hole, but I am going to tell you more about genes. So DNA, I've told you, is the blueprint for biology. Genes are the particular set of instruction to make proteins, and proteins are essentially biological machines that carry out complex biological processes. So pretty much everything that happens biologically are genes leading to proteins, and then proteins carrying out the construction of the biology, including building and sustaining the function of the human brain. Now, in order to do this with 20,000 component parts and building an incredibly complex organism and keeping it running, you need to be able to turn on and turn off genes in a highly, highly regulated way. And they generally work together in different sets in order to carry out these complex functions. So again, when you're thinking about the biology and when you're talking about genes, we are talking about just a little bit of a letter code, but it's actually a much more dynamic process than that. So we're going to think about gene activity towards the end of this lecture. And that activity, what I'm talking about, is the gene either being turned on or turned off to make a protein at a particular point in time and in a particular place in the cell or in the human body. So now genes, this expression, we call it expression, which is really a synonym for activity. If genes are working together, we infer that they're doing that and can demonstrate this with experiments, that when they're tightly working together, we call it co-expressed, that they're generally carrying out some sort of coordinated function. And I'm going to talk about gene co-expression later on. So those are the two terms from this slide that are most important to remember. So in order to give you a little bit of a visual sense of what I mean by genes being turned on and turned off, I'm going to rely on a video and a lovely narrator made by the Howard Hughes Medical Institute. And this is just a video of gene activity, and I'll let her explain.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands.
Okay, that's gene activity. And when you think about it, so you've got 20,000 genes in every cell in the body, and then it's exquisitely temporally and spatially regulated process that results in genes being turned on and turned off, and the genes then making these intermediate molecules called RNA, and the RNA then being turned into proteins. So that's the sort of dynamic process and of gene activity. And again, at the very end of the talk, I'm going to come back to this notion of gene expression. Again, you can hear the word activity when I say gene expression, and then co-expression when genes are working together. All right, so that's genes and DNA. I've told you about that. Now I want to give you one more concept that you can hold on to in order to get us through to the end of this lecture, which is genetic variation. So this is why I'm going to talk to you about genetic variation. Okay, if you turn to your left or your right, you are 99% identical with regard to your genome of the person sitting next to you. So I hope you all got a good seat or feel that you got a good seat today. So it is the case that with 3 billion bits of information, that regardless of where you look in this room, the person that you're looking at is 99% identical. as someone interested in genetic risk for disease, I'm not interested in the 99% that makes us all similar. I'm interested in the 1% that varies between people, because to the extent that genes are contributing to a disorder, they're contributing because of a change in the genetic code that's leading to some kind of function that's driving risk, in this case, the risk for autism. So in fact, I told you that the 3 billion bases was the easiest part of the problem because there are 150 trillion cells, or 150 billion cells, 150 trillion synapses. And then in fact, I don't even need to worry about all 3 billion bases. I need to worry about where the genome varies from one another, and that means that I'm now looking at somewhere in the neighborhood of about 30 million bits of information. Still not an easy thing to address, but beginning to be a tractable problem as a consequence of major technological breakthroughs about a decade ago. So genetic variation is what I'm interested in. When I say that I'm trying to identify a gene for autism, what I really mean is I'm trying to find a change in the genetic code that spells the letters for that gene that's different in a person with autism, that leads to that risk for autism. And there are different ways, different categories of genetic risk. What I just described is sort of the most commonly recognized, which is that the actual letter code of the DNA varies between individuals. It varies normally between individuals, and in some cases you can have a variation that leads or contributes to disease, in this case autism. But we also found, when we were able to begin to look at the genome in much higher resolution, starting about 15 years ago, is that when you look at chromosomes, if you have high resolution ability to look at actually the fine structure of chromosomes, and on this diagram it's showing you that a chromosome is actually just one long piece of DNA that's wound around like a solenoid and carries genetic material in the cell. So, but the structure of the chromosome can also vary. So we used to think that everyone had essentially the same structure of their genome, but only changes in the letter code of the genome. And then as we were able to look at the genome in high definition, what we found was that in fact we have spots where you might have 1 copy from your mom and no copies from your dad, 2 copies from your mom and 1 copy from your dad. So it transformed the dogma that you always had 1 copy of a gene from your mom and 1 copy from your dad. That change, if it falls below the resolution of a light microscope, we call it a copy number variation. That just means that we're looking at a variation in the number of copies of a small section of a chromosome. And again, that normally varies between people. You all have copy number variations. You all have letter changes in your DNA code. But it's a very small subset of those that contribute to disease risk of all types, including autism. The major reason that you differ in the 1% is where your grandparents came from and where their grandparents came from. Almost all of the variation in the genome is simply a consequence of ancestry. Small percentage, the small percentage we're looking for is related to disease risk. Okay, the last part of Genetics 101 is to talk to you about the difference between genetic variation that is transmitted from generation to generation and that's not. So most of the time when people get up and talk about genetics, they're talking about this intergenerational transmission of risk. So most people are used to thinking about genetics as something that's being transmitted, say, from grandfather to mother, from mother to daughter, and that, in many cases, people really think of it as synonymous with looking at the genetics of disorders. And the reason is the vast majority of variation that you have in your genome is being transmitted from generation to generation. But in each generation, in all of us, a very small amount of new variation occurs. And we call that de novo, which is just the word for new, Latin for new, de novo variation. So new variation in the genome, very small amount in every generation. It's a natural process that drives forward the diversity in the human organism, but sometimes it can hit a gene or another part of the genome that causes a problem. And in fact, de novo variation has turned out to be a very important place for us to look for genes that contribute large effects to autism, and this is why. If you have a variation in the genome that does something like contribute to a disorder like intellectual disability or autism or some other severe early onset disorder, natural selection is going to have a tendency to drive that variation out of the genome over several generations. If a person who has that variation is less likely than the people around them to reproduce, over several generations, that difference, even if it starts out being small, will tend to have the variations that contribute to things like autism and intellectual disability disappear in the genome. De novo variation, if you're able to see it in the first generation, natural selection has almost no time to act on it. The mutation appears, and the only thing that can happen is either the organism is born or it dies. If it dies, you won't see the de novo mutation, but if it's born, you can have very severe disorders, including disorders of brain development and function, that you can see and relate to a mutation because natural selection has not had generations to weed that out of the genome. So everything that I'm going to tell you about now in terms of gene discovery in autism has really rested on that hypothesis, that if we look at de novo mutation, this new mutation, it's going to be a place not to find all of autism genetics, but particularly to find genes that have large biological effects. And again, if you think about our strategy, which is find a flag in the ground, and then pull on the thread, that if you can find something that has big biological effects, that's going to be something that we might be able to turn into important new knowledge. All right, that's the end of Genetics 101. And now I'm going to take you through pretty quick history of where we are with regard to leveraging gene discovery, and particularly de novo mutation, to identify genes contributing to autism. So before 2000, the only thing we really knew about the genetics of autism was that there were some well-known disorders that were caused by a single gene known condition like Fragile X or other what we would have called mental retardation syndromes. And in those cases, kids were identifiable not just because of intellectual disability, but also because often they carry distinctive physical features. And in those cases, they're rare syndromes, we could see that there were increased rates of autism. But we knew essentially nothing about the common forms of autism, either actually of the children that you would have seen in the video. Neither of them had any distinctive physical features. Neither of them had a known developmental syndrome. So we knew essentially nothing about that. A key turning point was 2001, thanks to enormous investment in the Human Genome Project. Essentially what happened is we went from having no map at all and having to just sort of poke around in the genome in different sections of the genome. We got the complete map. It was sort of, the first version of a Google map of the human genome. And since then, it's just gotten better and better. And that was absolutely critical to allow us to be able to look across the genome and try to begin to identify changes in the genetic code that might lead to complex disorders like autism. By 2007, we had developed the ability to look at the genome in high enough definition that we could see changes that only encompassed about 10,000 bits of information. Now that sounds like a lot, but prior to that time, a light microscope, you would need to have something that changed 4 million bits of information to have any chance of seeing it at all, and often you could miss that. So this was an enormous increase in the resolution of being able to look at the genome. And when people use that to take a look at kids with autism versus without, the earliest papers in 2007 demonstrated that new mutations in the fine structure of chromosomes, copy number variations, were more common in kids with autism than without. Almost immediately after that observation, a wealthy philanthropist, and it's always surprising to make that shift and start talking about an individual, but in this case, it is an individual who literally transformed the field of autism science. So a man named Jim Simons had a family member who had autism, saw the early results with regard to de novo mutation. He's a brilliant mathematician as well as a wealthy hedge fund manager. And he decided that this was an important thing to invest in. And so he invested an enormous amount of his personal wealth to build a cohort to recruit people across the United States to take different academic institutions spread across the continent, have them all work together to identify families that would be particularly valuable to identify de novo or new mutation. Now, when you think about de novo mutations, it's essentially like having a lightning strike in the genome. It generally will happen once or less than once in every generation. And so what the Simons Foundation did, he created a foundation that was to create something called the Simons Simplex collection, and it consisted of families that looked like this. Blue means unaffected, and red means affected. And so the families were identified that looked like they had been hit by lightning, right? So they couldn't have any other family members that were affected with autism, just one kid. Now, this isn't how every family looks with autism. It's about 30 or 40% of the kids that see us. We get no clear history of anything that sounds like autism in the rest of the family, and then there will be an affected child. And so the notion here was that these kinds of families might help us identify these lightning strikes, these de novo mutations that were contributing to autism. He invested in creating a sample that included ultimately about 3,000 families, about 12,000 individuals, again, pulling together truly team science across the US. And I was very fortunate, as a relatively young person, to get a call from the foundation and to be asked to lead the genetic analysis of this group, the Simon Simplex collection. And pretty much everything I'm going to show you from my lab is a consequence of having the great fortune of being able to do essentially one experiment. And the experiment is simply to take the parents' DNA and the kids' DNA and to compare them. And remember, de novo mutation is a very low rate, so all I was looking for was very unusual events that would be present in the child but wouldn't be present in either of their parents. And that either meant that I was making a mistake, we were making a mistake, or it was a new mutation. And then the second thing that we were able to do, whatever tool we could use to look through the genome, we compared parents to kids. And then we compared kids with autism in the same family to kids without. Beautifully controlled experiment. We could ask, what is the rate of de novo mutation in these families? And then we could see whether or not, when you had essentially a 50-50 chance, if it was truly random, of having the de novo mutation land in the affected versus the unaffected child, what happened. And what we found is that the de novo mutation, much, much more often than you would expect by chance, landed in the kid with autism, suggesting that, in fact, we were replicating this early initial observation that de novo mutation was contributing to autism. The person up here, Stephen Sanders, who was a trainee in the lab, actually for many years, you're going to see his picture several times, very productive person. He's now an assistant professor at UCSF, and this is one of the first papers confirming at large scale, using the Simon Simplex collection, that these de novo copy number variations contribute to ASD, to autism spectrum disorders. And I'm going to focus on this title here because it points to one particular region of the genome as a particularly interesting finding. This wasn't the only one, but we pointed out that there was a particular region on chromosome 7, so all those letters and the letter and those numbers are simply a mapping coordinate. So on one of the chromosomes, the one that's numbered 7 in a particular region of the map, there was a small addition of genetic material, encompassed about 25 genes, and this reliably led to autism. And in fact, when we look carefully, it led to autism that would be indistinguishable from this young man, the video that I showed you. But we knew something already, just by being geneticists, which was that years before, the exact same region of the genome had been already characterized in kids, but in that case, instead of having one extra copy of this tiny section of the genome, they had lost a copy of this tiny section of the genome. And I'm going to show you what happens to kids who lose one copy of the same region.
Great, you're great, you got great hands.
Hi, Chris.
Hello, buddy, how are you?
I'm really watching ABC News.
Oh, thank you, thank you.
What's your favorite color and what's your favorite TV show?
Kids love to ask questions, but not as much as these kids.
My favorite color is blue. I have met Barney the dinosaur.
Yeah. Where do you live?
I live in New York City.
Do you have any sons or daughters?
I do.
Wow.
I have 2 daughters and a son.
And kids love to make friends, but not like these kids.
What nationality are you?
I'm Italian.
Okay, so every single one of the kids in this room has a loss of a section of the chromosome that's exactly the same as the section of the chromosome, literally exactly the same as the section of the chromosome that we found when you had an extra copy led to autism. And so what we identified here, we stumbled upon, was a region of the genome that has profound effects on the desire and the ability to have reciprocal social communication. And these kids have something called Williams syndrome, and they have many other problems. They have high rates of anxiety. Their social affinity causes enormous problems. They will follow strangers home. It's very difficult to teach them not to interact with other folks because the drive for social affiliation is so profound. So the reason that I'm showing you this is that when you think about the experiment that I laid out, actually it was pretty, you're swinging for the fence. You're saying, I'm going to take something as complicated as the interest and ability to communicate with another human being. I'm going to reduce that to one spot in the genome, and I think that I'm then going to be able to pull on a thread to understand something about the biology of something that complex. And this finding gave us a lot of hope that, in fact, we were headed in the right direction. Because if you do the thought experiment, if you knew which of the 25 genes in this region was leading to have such a profound effect, to be such a profound rheostat of social behavior, we would understand something fundamentally new about what supports and drives social affiliation, and potentially could figure out ways in kids who had difficulties with that to moderate that. Now, we love this experiment. It was definitely a happy moment when this was published, but it wasn't the exact experiment that we wanted to do. And the reason is from a biological standpoint, having 25 genes and saying one or more of those is likely leading to this problem is an enormously complex thing to try to dissect. So you don't really want to hone in on a region of the genome with 25 genes. What you really want to do is hone in on one gene and then begin to understand what that one gene is doing to simplify this problem even further. Now, 2011, we weren't quite able to do that. We were just on the verge of doing it, and this is why. So the Human Genome Project not only led to the creation of a map, it also led to a technological explosion that has driven down the cost of looking at every base in the human genome by a remarkable amount. That's Moore's Law. I'm from Silicon Valley, so this always impresses the IT folks I show this to. Moore's Law is that line, and then the green is the rate of decrease in the cost of sequencing a human genome. So what this did is it allowed us to go from looking at 10,000, 20,000, 30,000 bits of the genome simultaneously to looking at every single letter in every protein in the genome simultaneously in every patient that we were looking at. We ran exactly the same experiment, comparing the DNA of the parents to the DNA of the kids. We looked for the rare event that was present in a child that was not present in a parent, and then we asked, is that more likely to land in a child with autism than to land in their unaffected sibling? And just like copy number variation, it was, with the little twist being that if that variation caused problems for creating a protein, you would see an increased rate in kids with autism. If it was predicted to do nothing, that the protein was still able to be produced normally, then we saw no difference, which is the perfect control. What that tells you is it's not just that you're randomly getting more mutation in a kid with autism, it's telling you that the mutations that damage proteins are a cause of autism in the kids that you're seeing it in. So by 2010, this technology was developed, fortunately, right across the hall from me at Yale, so I got to use it very quickly. We applied it to autism. By 2012, we were able to show that these sequence changes in autism were doing the same thing as those sections of the genome, and we were able to identify the first clear-cut genes for idiopathic autism, where there was no argument about whether or not it was contributing. Other labs were stride for stride. When we published in the journal Nature in 2012, there were 2 other articles in the same issue, one from Boston, hate them. Sorry, I hope they're always there. And another from University of Washington. I don't really hate them. So we also saw some very interesting things. The first was contrary to sort of the mythology that had grown up in psychoanalytic theory that mothers were the cause of autism because they lacked empathy for children. We found that the vast majority of these new mutations in sequence were on the father's chromosome. Thank you. This is for a particular type of mutation that contributes to autism. I'm going to tell you about how much. It's not all of autism, but these were clearly on the paternal chromosome. And moreover, we could tell that one of the reasons that they were accumulating is that sperm continue to reproduce throughout life. And consequently, as fathers got older, they naturally will have more de novo mutations. And those de novo mutations, a small proportion of them, are going to contribute to autism. This begins to give you some insight into how you can think about a a largely genetic disorder that's changing over time. Because simply having people have a tendency to get married later and have kids later over 40 years could contribute to an increasing rate of autism and shows an interaction between a strongly genetic disorder and an environmental factor, in this case simply age, but you can imagine that this would also be a way to begin to tease out what other environmental factors might contribute to de novo mutation and therefore contribute to autism. By 2014, a number of other labs, and particularly large sequencing consortia, some funded by the Simons Foundation, importantly, huge contribution from the National Institute of Mental Health for an autism sequencing consortium. Dozens of genes with large effect have been identified for autism. And we can predict that there are going to be between 500 and 1,000 of these ultimately when we complete the experiment. All told, this type of mutation accounts for about 25% of the kids that we see in clinic. Now, it means that there's a lot that we still don't understand, but again, the strategy here is just to gain some insight into the underlying biology. The bottom line right now is that within that 25%, we understand less than half of that. And consequently, what we know is that if we continue to recruit families and we continue to sequence them, we will continue to discover genes. It has become essentially a rote process now. We can essentially, if there is a will to, we could uncover the entire range of genetic mutation that's contributing to autism via de novo mutation using the methods that we've developed. This is an article in 2015 that shows the sort of the state of play in the field. This is still pretty current. This is a table that shows all of the genes that have been identified. Those are just genetic abbreviations for each of the genes. Again, you can see that Stefan, as I told you, remarkably productive trainee in the lab. And the blue box there are the genes about which there is no longer any debate. These are definitive autism risks. They carry very large effects. If you carry one of these, on average, you would have 20 times the risk of developing autism compared to a child who did not carry this. And all the rest of these genes have genetic evidence, and simply a matter of doing more and more sequencing to weed out the ones that are true versus a very small number that will be false positives. Okay, so I'm going to finish by giving you a 30,000-foot view now of what we have learned from this and just a general notion about what we think we can learn. So the first thing that I want to say is that genes, we know enough about genes to assign them roughly into categories about what they do. Now, most genes do many things, so this is not a perfect way to try to divine what the genes are that we've discovered for autism are doing. But as a general kind of bit of analysis, you can simply say, what do we know about the genes that we've already discovered? This is a picture of where two nerve cells connect with each other. It's called the synapse. And one of the first things that we were able to see without even doing any fancy analysis, that's a blow-up version of the proteins. Now, each one of those proteins is made by the gene of the same name. And all of those proteins contribute to how these neurons are communicating with each other. And the first thing that we could tell, even without statistical analysis, we had a boatload of genes that were contributing to this transmission of neural signals at the synapse. So there are other biological processes, a handful of them, that had the same characteristics. We could just look at the genes and say, it looks like the genes are involved in regulating other genes, and it looks like the genes are involved in synaptic function. Now, the more that we understand about these genes, the more that biology moves ahead, the more we'll be able to take a list of genes that we have for autism and say, what do they do? But the second thing that I want to describe for you is thinking even beyond that. So this is a developmental disorder. So you might ask one question, what are these genes doing? But another very important question, if you wanted to do something about autism, would be to say, when and where are these genes having their activity? Now, that's a much more complicated question, but actually we thought, starting in about 2012, that we might be able to gain purchase on that question. And the reason that we thought that is just down the hall from me was the gentleman on the right, wait, what's on your side? It's your right, yes. And the person, the mustachioed gentleman is another trainee in the lab, Jeremy Wilsey, again, a brilliant, brilliant trainee who helped lead this project. But Nenad Sestin, the gentleman with the glasses, was a junior faculty member just down the hall. We were good friends, and he does basic neurobiology. So the question that he wanted to answer was not about disease risk. He wanted to know, how does gene activity build a human brain? And so what he did is he got a whole bunch of human brains shortly after people died of natural causes. And this, actually I say people, but it was fetuses all the way through 80-year-old people, 4 weeks of age to 80 years of age. And he cut the brains up into small, anatomically distinct regions. And he looked at all of the expression, at all of the genes in all of those regions. And he began to build, for the first time, a map of the trajectory of gene activity across human brain development. So I knew he was doing that down the hall. That actually turned into another really important large-scale initiative called BrainSpan, and that's being funded by the National Institute of Health as well as the Allen Institute. And this is to build really the first map of the activity of genes in human brain development. So it started down the hall from me, and the two of us were talking. We thought, you know what, this is Reese's peanut butter cup. I've got chocolate. You've got peanut butter. I've got genes that I know lead to autism, and you've got a map that can tell me when those genes are active. You can also tell me when they're co-expressed. Now, remember I told you co-expression, I was going to come back to that. So when genes are co-expressed, they're working together, and they're working together in space and time. So we looked at gene co-expression, and we asked the question, if we took our autism genes and we took this map of the molecular landscape of human brain, could we find a co-expression network when genes were working together that carried many more genes for autism, meaning they had much more risk than we would expect by chance? We were able to find that. Shockingly, we thought we would look all over the place, but in fact, we found one network from the initial genes that we discovered that was dramatically enriched for autism risk. And that network we were then able to ask questions about. We could place it in human brain development. It turned out that that network was really only present and working together in mid-fetal gestation. And not everywhere in the brain, but in a particular region of the brain, the front part of the brain called the cortex, and the prefrontal cortex, which controls kind of high-order human cognition. So we went from genes to understanding something about where in development they were having their effect, to understanding a little bit about what region of the brain, and we could even go further than that. This particular signature, the genetic signature from this gene expression, was only restricted to 2 of 6 layers of this multi-layered structure called the cortex. So now we weren't in the cortex generically, we were in what are called layers 5 and 6, and those layers are the layers that send long-range projections to other parts of the brain and make synapses, the other kind of biological clue that we had, with their very first synapses that help control linking the front part of the brain to other regions of the brain. Now, this is still a story that's unfolding, but the point here is that we were able to move from gene discovery to begin to tell you something. I mean, I have to say, it's, it probably just seems like this is sort of, ordinary lecture. We wandered in the dark for 20 years. To be able to stand up here and tell you, I've got dozens of genes for autism. I can not only tell you what they do, I can begin to tell you when and where in development they're relevant, is a remarkable thing. And remarkably important, I think, and I'm going to conclude with this argument. So we can now reliably, reproducibly identify autism genes. More families will mean more genes. Equally dramatic advances right now in understanding neuroscience and computational sciences are giving us a substrate just in the way that I described with that last experiment, but in other remarkable ways now as well, to take those genes and place them in the context of human brain development. And that investment in basic science is giving us the context to understand disease risk. Often you'll hear debates about, should we fund basic science, should we fund translational science? In this case, without the combination of the two, the gene discovery is going to get you nowhere. And just understanding the basics of brain development is not going to tell you about why people have autism. You need to have both. Take home, breakthrough technologies empowered by investment, largely from folks that you work for. Empowered by the Human Genome Project. Breakthroughs in gene discovery as a result of de novo mutation. Almost all the progress in the genetics of autism has come as a consequence of looking at this subset. We've gone from wandering in the dark now, being able to begin to tell you what, when, where, and how. This has happened as a consequence of effective advocacy by parent organizations and patient organizations, philanthropy like Jim Simons, Autism Speaks, critically important NIH funding, and people working together. The labs could not do this independently. If you saw every article that I showed you out of cast of thousands in the author list, it can't be done in one lab at a time. And now we have major challenges. And I realize that people say, well, what has genetics got you lately? How is this really going to lead to a treatment? And what I tell people is we're exactly in the same spot that people were at with cancer 30 years ago. It went from this big, broad diagnosis that no one understood to the first molecular clues, and from those first molecular clues now to transformative therapies in many areas of cancer, things that we never could have imagined without understanding the biology. Transformed life expectancy, particularly for pediatric cancers. And I think the same kind of optimism, it's going to be a long road, but we have now the substrate, and there's a clear path forward to do the same thing with autism. So hopefully I've gotten to the end, which has started at the beginning. The genetics has already transformed the understanding, but continued support is going to be critical. And because I've got you in the room and I read the instructions, said if you have a pitch, you got to make a pitch because you guys, have influence with people who have, make policy, is I just want to say that this is autism, incredibly important, been very fortunate to have it well funded. That's not the only story. I work with other kids who have serious neurodevelopmental disorders. This is, in fact, the story that I've told you. You could take out the word autism and say neurodevelopmental disorders. For Tourette syndrome and obsessive compulsive disorder, we're about 5 years behind autism solely because of an absence of funding. Just in May, that's the cover of Neuron, paper from my laboratory showing we can do exactly the same thing. We can find genes as reliably now for Tourette syndrome and OCD. We know we can do the same for epilepsy, for intellectual disability. It looks like for attention deficit hyperactivity disorder. There's a path forward now to understand things that were really, truly mysterious not a decade ago. And I hope that I've convinced you that this is a worthwhile thing for us to be doing. Thanks for your attention. These are all of the folks that contributed. Actually, they're not all the folks. This is a slice of hundreds of people, really, and the most important folks that I have to thank always is without the patients and families at Yale, UCSF, and the Simons Foundation contributing, I would have nothing to tell you. Thanks for your attention.